Welcome back, fellow mitochondriacs. I had the distinct pleasure of interviewing Professor Dom D'Agostino from the University of South Florida from the Department of Molecular Pharmacology and Physiology. I had an amazing conversation with him, and I hope you will get benefit from conversations like this because these individuals are absolute experts in their field, and these are the type of folks that are going to help move this movement forward. And I hope you can see the value in that. I know that I certainly did. So without further ado, let's get into it.
I've done.
So again, thank you, Professor Dominic D'Agostino, for being on our show. I appreciate your time. And I just wanted to ask if you would do a quick intro to how you ended up where you're at.
Sure. Yeah, my background is undergrad nutrition and biology, and then I studied neuroscience and physiology for my PhD in a pulmonary critical care division department. So I had in a clinical department, like working, with residents and fellows, actually trained them on research as a PhD student and taught like grand rounds and stuff. And then I did my fellowship, my postdoctoral fellowship funded by the Office of Navy Research specifically on oxygen toxicity seizures. So the neuroscience of seizures and preventing, uh, essentially like trying to understand cellular and molecular mechanisms of oxygen toxicity seizures in a warfighter performance program designed to enhance safety, resilience, and performance resilience. I guess you could call it that in extreme environments. So, as you know, special operators are in extreme environments. It's preventing them from enhancing their performance and also safety mitigation, especially from a neuroscience perspective.
And my understanding is, sir, that you were at one point, I watched a podcast where you were talking about how you were doing basically like these Navy SEALs were going to like extreme depths and they were having seizures that were not mitigated by anti-epileptics. And you were saying that ketogenic diets and ketones were able to help mitigate that. Is that my understanding?
Yeah, we don't really, well, that my research is fundamentally understanding oxygen toxicity seizures. And with a closed circuit rebreather, which is used by special operations and some, special operations missions, it is closed circuit, so there's no bubbles, so the concentration of oxygen you breathe is high. And according to the dive tables, is the potential for oxygen toxicity exists with 100% oxygen at just 50 feet of seawater within 10 minutes. Really. So these happen. They're rare, but you have situations where a diver needs to go down and plant a mine at, 60, 80, 100 feet. And that's like, the potential for oxygen toxicity is quite high. And oxygen toxicity is the limitation of hyperbaric oxygen therapy, which we can talk about. And there's 14 different FDA approved applications. And I like to, we like to do things, we're like on the edge of science kind of, and we kind of like push the limit. So I always had this sort of idea that if we can go from 2 or 3 atmospheres, to 5 atmospheres and actually make that safe, then we could like quickly reverse carbon monoxide poisoning and save a patient. We could quickly reverse gas gangrene. We could quickly mitigate decompression sickness, or as I got into cancer research, we could basically blast tumors that overproduce oxygen-free radicals with like 5 atmospheres of oxygen, if we put them into a high state of ketosis,
Mhm.
because that will prevent. And then you have the potential for pulmonary oxygen toxicity. So we work on that front too. But yeah, we're funded by Office of Navy Research, and NAVSEA, which is part of the Department of Defense. I also work with Special Operations Command on different things. Now we're working on a TBI thing, and also we've done some work with DARPA in the past too. So
That's not
most
in
of,
your neck of the woods, right? Special Operations Command is around Tampa, right? Like in that
oh
area.
yeah, 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 we're working with them and on different fronts now, and yeah, they have like maybe less funds than the Office of Navy Research, but their our work is very in line with sort of and we have very unique equipment in our lab too, which allows us to explore extreme environments.
Awesome. Okay. Well, I'm going to assume that you probably have had this, but I'm just going to ask, when was it when you had, that I'm assuming, the aha moment that cancer is a mitochondrial metabolic disease?
Yeah, it was a gradual transition and curiosity because I was trained as a neuroscience and I'm studying oxygen toxicity funded by the Office of Navy Research, and we developed some unique technologies that allow us to look at different cells, tissues, animal models, ranging at different models, and also now we do human clinical trials with some of the things we developed. But I think it was the observation that I basically, designed something called hyperbaric atomic force microscopy, and then later got another grant to expand that to laser scanning confocal microscopy, which allows us to look at the mitochondria. And in the process of, creating the fluid, gas, and electrical penetrations for this equipment, and then ultimately testing it to show that we could have like sub-nanometer resolution at high pressures of oxygen, I did a lot of different cell types. And one cell type that was very easy to actually study was a human brain cancer cell type. I believe it was taken from a 44-year-old patient. It's a U87 glioblastoma cell line and a few other GBM cell lines. They're just so easy to grow relative to primary, dissociated neurons, which I
Hmm.
spent like 10 years like growing them on plating them on astrocytes. And, it's almost like voodoo sometimes to get really good cells, but cancer cells are easy. I mean, they'll just grow
Hmm.
like on top of the bench top. So we had these cells and we had muscle cells and everything. And we were exposing these cells to graded levels of oxygen, and it was very clear to me that the cancer cells are like super robust, and it's like hard to kill them. But under normal conditions, like in the lab, it's an easy model to work with, these immortalized cell lines. But I realized I observed that they were dying fast under a level of oxygen that was relatively non-toxic to primary dissociated cortical neurons and hippocampal neurons and also brainstem neurons. And that piqued my curiosity because I did not know a whole lot about cancer. I was a little reluctant to go into cancer research, but I just reached out to Moffitt Cancer Center and I just started digging in, like, why are these cells dying, and could that be something interesting to like study,
Hmm.
right? Because I did quite a few PhD students were entering the lab and they want to do PhD projects, and one was Angela Poff at the time, and or Bennett at the time before she got married. And Dr. Angela Poff now, she's my research associate. She wanted to study, these cancer cell lines. So we had the idea of using hyperbaric oxygen with a radioactive conjugated nanoparticle. So the idea was that, because these cells are dying faster, and the Moffitt was telling me that the efficacy of radiation therapy is proportional to the PO2 of the tumor. So the idea then was to bring the radiation to the tumor with a chlorotoxin conjugated radioactive nanoparticle that would decay in a couple hours. So, not to throw too many big terms out there, but to take something that would bring a pulse of radiation to the tumor in a predetermined time, and that patient could get inside a hyperbaric chamber, and this particular molecule just attaches onto glioblastoma, and it would like pulse the radiation while you're in the hyperbaric, oxygen
Hmm.
chamber, and you would have to like radiate through the brain. So, we put this idea into different grants. It was like reviewers are like, wow, this is really cool, but you got to show us some preliminary data, which is always the case, right?
Hmm.
So that didn't get funded, but I got more interested in ketones and ketogenic diets because these were preventing seizures. And I was like, oh, with brain cancer, well, these patients are often put on like, dexamethasone and anticonvulsant drugs. Well, why don't we just put them in ketosis? And then there was a 2008 paper that came out of Dr. Thomas Seyfried's lab, and it was like in a CT2A model of brain cancer, and he had written a couple other, and then there was by the author Nebling had a case series report of using in pediatric brain tumors a ketogenic diet in pediatric patients. So this caught my attention using a ketogenic diet. And I was like, wow, maybe we could, do a project of using a ketogenic diet in a brain tumor model and then couple that with, since this seems to have some anti-cancer effects, we could couple that with hyperbaric oxygen. And then I had some ideas. We were using lonidamine, 2-deoxyglucose, and a few other drugs that were killing the
Hmm.
cancer cells. And that was like little pet projects I would put medical students on. So that was 2008 or nine, I think. And by about 2010, I took on some graduate students and I assigned them to the projects. And I think it was the aha moment was the validation. I reached out to Dr. Seifried and he wouldn't give me the brain tumor model, but he would give me the VMM3 model of metastatic cancer, which I could conceivably put those cells into the brain and make a brain tumor. But he was like, this model of metastatic cancer is unlike anything anyone has ever seen. I think it had just made the cover of International Journal of Cancer. So that was like, okay, this is a pretty legitimate model. And I liked it too because the animal, it's good in the way that the animals die kind of quick, like within 3 weeks, they
Hmm.
get such rapid cancer growth and metastasis that they usually succumb to the tumor burden and die. So I was like, okay, this is a good way to vet out whether this is going to work. So he sent us, the cells which have a gene in them to make luciferase, and you inject luciferin, and the cancer cells bioluminesce based upon, making this luciferase enzyme. So you could, without using like a PET scanner, you can actually image
Right.
the bioluminescence of the tumor growth, and that was really cool. That was another, and it was like a syngenic model, like it was an inbred strain. The animals were not immunocompromised, which is a whole nother problem with these animal models. It seemed to like overcome, like, we've cured cancer in mice,
Hmm.
but there's problems with the models. And this
Right.
model, kind of like that he developed, seemed to overcome a lot of these limitations. So we embraced the model and it worked remarkably well with a ketogenic diet. And then we did ketogenic diet and hyperbaric oxygen. And then we did ketone supplementation with we did ketogenic diet plus ketone supplementation plus hyperbaric oxygen. And that worked really, really well. And I think we had results unlike anything that he maybe had ever seen. And later, I think he did some work with Dawn that rivals that, but it's pretty close.
Hmm.
Um, and yeah, and he had done some work, I think, using the model with chemotherapy, maybe methotrexate, maybe cisplatin. But I mean, we went beyond, those results too with the survival. So that I was a very skeptical, but because of my background, I didn't know that much in talking with people. They were not super convinced of the cancer as a metabolic disease theory, but the more I read about it and the more I think it was more experiential, right? So I have a background in sort of biochemistry and cell biology and everything. And what he was presenting made a lot of sense. And there wasn't a whole lot of arguments against it. Like we would have lab meetings and be like, okay, critique this theory. And then we would do like roundtable. It's kind of hard to critique the way he was presenting it. At the
Hmm.
same time, I was also curious why other investigators were not like, I don't know, embracing it and trying it. So I did not have funding to do this research, but I did get a little bit of startup funds when you transition into a tenure track position. I had like maybe 50K of startup funds, and we were able to limp along on that. And actually, I think, and then we got like some little grants through industry, and maybe in $100,000, we like published 3 papers like out of that. And that was pretty game changing for us in regard to changing my mind that cancer is a metabolic disease. It left many questions, in my mind, like how, we did like the proof of concept, but then it was just like, okay, what about dosage, timing? What about other forms of cancer? What about,
Hmm.
standard of care? Can this be synergized with standard of care? And I didn't really want to tinker around with radiation and chemotherapy drugs in my lab. So we made a commitment early on to focus on non-toxic. But well, there's some relatively toxic drugs. So I guess I would say not using chemotherapy and radiation if we didn't have to. But we've done with like metformin, we've worked with that, sodium phenylbutyrate, 2DG, lonidamine, and a few other drugs. DCA, I had a student just did a whole PhD project on DCA. Dichloroacetate
Hmm.
was relatively effective too in the model. But nothing is, the ketogenic diet is the biggest lever. And then ketone supplementation too, even integrated into a standard diet, actually had pretty remarkable effects
That's
uh,
interesting.
because by changing metabolic physiology.
Fascinating. Yeah, I didn't want to cut you off because you're on a roll there for a second, but
Yeah,
I was like,
sorry, I
yeah,
could ramble.
it seemed like it was more the fact that you were experimenting and getting it that way, not so much like the mechanism or the, like the looking at Warburg stuff. And I mean, it seemed like it was more that the things that you were getting out of experimentation, that was what really kind of pushed you over the edge. Is that fair to say?
Yeah, for me it was just outcomes-based research. I was like, well, let's see if the animals live longer, and then once we establish that, I'll be interested enough to dive into the mechanisms. So, I mean, over the last 10 years, I've only studied mechanisms. So we've published, beta hydroxybutyrate suppresses the NLRP3 inflammasome. We studied beta hydroxybutyrylation, which functions, BHB functions as an epigenetic regulator. We've looked at, GABA levels in response. We do a lot of neuroscience, ketone metabolic therapies remarkably change the neuropharmacology of the brain in a way that can prevent seizures, mitigate consequences from traumatic brain injury. So my wheelhouse has really been mechanisms, but I think we kind of start top down. Like we do the research to figure out, what's going to enhance survival, and then unlike the the NIH model, which is like you find a mechanism and then you kind of,
Yeah, I
from a
get that.
very reductionist point of view, kind of work up towards, a therapy. We did the back end of it, and we've done
Gotcha.
that with ALS research. With the Deanna protocol, there was a patient, Deanna Tadone, still alive today, was only given a few years to live, well over a decade ago. And the Deanna protocol is a metabolic-based therapy for ALS. And it
Interesting.
incorporates, AKG and idebenone at the time and GABA, and then some MCT2, and that's pretty remarkably effective for ALS. But it was like, the patient was there, and I met with the patient and the family, and then that funding went to the animal model, and now we just, look at mechanisms
Got
from
it.
there. So
Yeah, ALS, what a terrible, terrible disease.
yeah, that one's a hard one. Yep.
It's tough. That may be worst case scenario, especially because it's not, it's kind of like the opposite of dementia where your brain is gone, but your body can potentially be intact. But when your brain is intact and your body goes, that's rough. That's tough. That's a terrible thing.
Yeah, well, I can tell you that metabolic-based therapies are above and beyond, the standard of care. There's a few websites that actually evaluate these things. The first one, I think, was Patients Like Me. So if someone's interested in ALS, just go to the website Patients Like Me, put in the standard of care, which is Riluzole, right? And then look at the feedback on that, and then put in the Deanna protocol. You get like, the patients, there's about three times higher positive reviews,
Wow.
proportionally on the Deanna, but nobody knows about it. We've published maybe 3 papers on it, but
Hmm.
we have not moved that into an RCT, randomized controlled trial. But yeah, I've been in touch with enough patients to know that, these metabolic therapies work in many different domains, from seizures, which was the original application. And when I connected with Thomas Seyfried, he said something very interesting to me because I was only focused on seizures at the time. And I
Hmm.
read his paper, Cancer as a Metabolic Disease, or the 2008 study, and I got his email, and he answered the phone, and we started talking. It was quite surprised, and we had a lengthy conversation, and I was just telling him my results. And he made a comment that really uh, sparked my interest. He was like, "Oh," and he said it in a very, not an authoritative way, but in a very confident way. He said, "Yeah, well, ketogenic therapies are more effective for cancer than they are for seizures." So he's like, "Yeah, you might want to think about,
Hmm.
pivoting from seizures to," and that really caught my attention because I was only familiar with the seizure literature. But in 2010, or well, I started actually in 2005 or six, just tinkering with these cells and just as like little pet projects. But it wasn't until like 2010 where we actually started doing like animal model research,
Hmm.
validating it. And
It's kind of funny. He started with seizures too with ketogenic diet.
yeah, he did. Yeah, he was actually chair of the American Epilepsy Society Special Interest Group at the AES meeting, which is more like a pharma meeting. And I later became the chair for like 3 years of the special interest group in ketogenic diets. And then I think my research associate, Dr. Poff, did that too. So yeah, we've always been kind of had that background in seizures, and that's where you learn a lot mechanistically about how ketogenic diets are neuroprotective and have anti-seizure. And you learn a lot about mechanisms in regard to insulin suppression. So the keto, a lot of people talk about glucose, but I think of ketogenic diet as insulin suppression therapy, mTOR suppression, IGF-1 suppression,
Hmm.
limiting glucose availability, elevating ketones, and then hitting we look at antioxidant defense mechanisms like the pentose phosphate pathway and some like glutathione, and these tend to go down and they make the cancer cells selectively vulnerable. So
Hmm.
you kind of learn a lot about the mechanisms, and when you learn about cancer metabolism, growth, you realize that you can leverage, this metabolic state, not to cure cancer. I like to be very clear about that, but to create a weaker target for other modalities.
Correct.
And I don't think we've actually ever studied the ketogenic diet in and of itself. We've always done the ketogenic diet as like the base therapy to test other
Well,
modalities.
I think that's how it should be. I think looking at ketogenic diet by itself or even Dawn by itself is, I don't want to say stupid, but it doesn't make sense. It makes
Yeah.
sense more of in a systematic way. So whenever you only see one intervention at a time, and that's how it's done in science. They want to look at one variable, and I get that. But
Yeah.
when you're talking about like this, it makes sense that it's done as a system. And that's
Mhm.
where I would love to see a trial come out with that. But it seems like that's going to be very difficult to get. Um, what can you give me, I don't know, like some of the less well-known things that ketones can do, endogenous ketones can do to fight against cancer?
Yeah. So with that, I'd like to maybe just pivot a little bit and just talk about like prevention,
Oh,
right? And I
sure.
think,
Sure,
yeah.
sure.
Yeah, I'm just going to mention that like real quick because we just presented some work to the at the med school, and there was a big review article that came out in and it's the journal the basically the flagstaff, the flagship journal of the American Cancer Association, and it's a over a 500 impact factor. So this is like really, I think it's the highest impact factor of any journal. And the title of it is the proportion and number of cancer cases and deaths attributed to potentially modifiable risk factors in the United States.
Hmm.
And it was pulling from data from 2019. And I think that an important message is that also, in line with psychiatric disorders now in a field of metabolic psychiatry, is that our metabolic health and our metabolic state is a big driver for cancer. And I think that cannot be understated. And we know that obesity is like a huge driver for that. So 50% of all deaths in the United States are linked to modifiable risk factors. So that's something to consider. Metabolic health is tied to 9 out of 12 of the modifiable risk factors for cancer. And then, this was not even mentioned, 50 years ago, right? Like nobody talked about metabolic health. And even like the non-diet or metabolic health related factors, modifiable risk factors that are associated with cancer, even those you could talk about ultraviolet radiation, you know, uh, uh, hepatitis B virus, I think, Epstein-Barr virus, CMV, HPV, for example. Like these viruses actually cause cancer by modifying the genome to some extent, but these viruses also specifically like laser target the mitochondria. So the mitochondria are easily damaged, and when you accumulate mitochondrial damage, the DNA repair mechanisms in the mitochondria are far, far less robust in repairing that. So we have very robust DNA repair mechanisms at the level of the nucleus,
Hmm.
whereas mitochondrial DNA repair is not very good. So you get progressive inflammation, you get virus-induced mitochondrial dysfunction, and it's more of a protracted kind of thing, right? Like with inflammation and with the virus is attacking that. And the bioenergetic state of the cell, the ATP production, when that gradually declines, the retrograde response, like the nucleus can sense the energetic state of the cell. When it
Hmm.
does that, sometimes it dies, but if it survives, it then kicks on oncogenes. And many of these oncogenes are genes that enhance glycolysis. So oxidative phosphorylation will progressively decline with many of these provocative agents that cause cancer, right? That's the oncogenic paradox. You have many different things that can kind of cause the same thing.
Hmm.
Uh, and I think, the important thing is that it just sort of feeds into the cancer as a metabolic disease, not only as as a way to treat it, but it like ideologically, it is.
100%.
So, and that flies in the face of what a lot of people have learned. But I think science is being rewritten, and I think there's a greater appreciation for specifically the mitochondria. Very robust, healthy mitochondria are the ultimate tumor suppressor.
Hmm.
So I think that you can kind of put it in that way, and that's why that's why we don't really get the mitochondria in the heart are super robust, right? We look at like cardiac cells, the mitochondria in the muscle is super like robust. In other areas, it could be damaged more easily, right? With progressive, like the liver, for example, with like hepatitis B virus, or you have these organ-specific viruses that affect certain organs and cause chronic inflammation, and that inflammation too is a major driver for mitochondrial
Hmm.
dysfunction. So, not to like sidetrack the conversation, but I think there is no cure for cancer right now. We're working on that, and Thomas Seifert is sort of spearheading projects that I think will, and I'm really honored to be part of it, spearheading projects that I think will change the standard of care. But I think that prevention is largely the cure when it comes to cancer. And if we appreciate and understand cancer as a metabolic, genetic, and epigenetic disease. That's
Hmm.
how I like to kind of frame it when I'm teaching like the students. But I would actually centralize the metabolic control of epigenetic regulation. So that's something that we study in the lab. Also, the fidelity of the nuclear genome is intimately linked to the bioenergetic state of the cell. So the capacity of the mitochondria to produce ATP and preserve the robust DNA repair mechanisms and the bioenergetic state of the cell will basically by default prevent that cell from the transition from a healthy cell to like a cancer cell. So that I think there's good evidence to support that with some of the research. That's essentially the essence of like Thomas Seyfried's book, and also what Otto Warburg presented is that respiratory insufficiency is the kind of the hallmark characteristic of cancer cells, right? And respiratory insufficiency being not
Oxphos.
respiration, but like oxidative phosphorylation,
Right.
and that's a hallmark characteristic. And maybe he was not 100% right about a lot of different things, but he was way ahead of his time.
Yeah. It
And I
doesn't
think
seem like,
moving
though, when you
forward,
read a lot of papers, though, that does not seem to be permeated throughout the literature. I mean, it seems like a lot of investigators believe that oxphos is the primary energy production for a cancer cell. And
mhm.
it's just hard for me to understand because I talked to Seafried about this saying, and we laughed about it because it says that because you'll read the most malignant cells are in the most hypoxic region of the tumor, but then they'll say that's using oxphos, and it doesn't make sense. Like why would these malignant cells that are in these hypoxic regions be able to use oxphos as their primary way of producing energy? I mean,
Well,
aside
they
from the
consume
mitochondrial damage that
oxygen.
is there, right?
Yeah, they do consume oxygen. I could say that just by, studies using the seahorse biosciences machine, that they consume oxygen, but the oxygen consumption is not coupled to ATP production in the same way the healthy cell would be, right? So, and I think that defines it. And you also have oxygen consumption. also generating a lot of that oxygen is then goes, which was our first observation, is that, if you push more oxygen through the mitochondria of cancer cells, they produce proportionally more superoxide anion.
Hmm.
And we could leverage that in ways to make that superoxide anion more reactive intermediates. So you get, superoxide anion goes to hydrogen peroxide, then hydroxyl radical, and then there's other sort of nasty things.
Peroxynitrate and stuff like that.
Yeah, epoxy that you can generate from that, and cancer cells will upregulate their antioxidant defenses. And I spent 5 to 7 years studying heme oxygenase. So heme oxygenase 1 is upregulated in that. So you could potentially use like a heme oxygenase inhibitor. It's probably one of the central defenses that cancer cells use. And they also sequester a lot of iron. So, and this is the porphyrin overdrive hypothesis. So cancer
Mhm.
cells suck up a lot of iron. A neuro oncologist or neurosurgeon will use something called 5-amino levulonic acid, or 5-ALA, and they give 5-ALA. The patient consumes it, and then they go to do surgery. They do that because 5-ALA is the precursor to heme. So you'll generate a lot of heme, and then the cancer cells will suck up massive amounts of heme relative to the surrounding healthy tissue, and then you could. With a light, you can it fluoresces, so you can site specifically pull out the tumor cells. Theoretically, you could, so that actually generates a lot of oxidative stress too. So
Right.
you could,
Photodynamic therapy, basically.
yeah, and one idea could be, 5-ALA, a heme oxygenase inhibitor, and hyperbaric oxygen therapy. Like this is something like,
Hmm.
that I've discussed, I've told oncologists about, and they're like, that makes all the sense in the world. Like we should do it. We got to write a grant to do it. Like these are things that I'm just throwing this out there. Hopefully someone will pick up on that. Or even like patent it, or like do something, just move it into clinical trials, because
Hmm.
everything that we've done in regard to like little pet experiments basically shows that what I just described would be like largely non-toxic, not toxic at all to healthy cells, and 100% fatal to glioblastoma cells. So if you hit them, and then you could take it another step further and use something like artemisinin. So
Hmm.
artemisinin like kills malaria through an oxidative stress mechanism, right? So hyperbaric oxygen, artemisinin, heme oxygenase inhibitor, in the context of a GKI of 1 to 4, I think by all stretches of the imagination that will work far greater than anything that we have, right? And what I just described is relatively non-toxic.
Right.
And a patient could conceivably just get these things online and
And they
use
do.
it themselves, right? So
They
yeah.
do.
So
Yeah,
5-ALA even. I
that's
mean,
actually
artemisinin you can get.
interesting because that's the way I've thought about it, is kind of like a three-legged stool. Basically, like the glucose and glutamine, yes, bioenergetics, ATP, and I'm sure that just just hitting that probably, and again, I'm not a scientist, so I don't know, but my guess is that if you restrict glucose and glutamine to sufficient amount, that enough would collapse the bioenergetics of the cell, and then you couldn't have any ATP dependent processes. You couldn't probably have any redox biochemistry because you'd have no energy to support that. But that being said, I see glucose and glutamine as really the 1-2 punch before you hit it with an oxidative therapy, because then you have no ability to, or a less ability to build glutathione and recycle it through NADPH with the PPP, right? So it just seems like that would be even what you just said, which sounds awesome, basically photodynamic therapy with the brain, with hyperbarics, et cetera. Like, but it still wouldn't make sense to me unless you had the other two things adequately covered first. Does that make sense to you too, or no?
Yeah, that's the idea. Get the patient, transition them into with ketone metabolic therapy, and use a combination of a modified supplemented ketogenic diet. It's a little bit higher in protein. You supplement that with medium chain triglycerides, also with a an alcohol-free exogenous ketone, meaning no butanediol. So pretty much every ketone ester or precursor on the market is 1,3-butanediol based. That could work, but we have some evidence that that could be toxic. So my preference is to use a ketone electrolyte salt. specifically use racemic,
Hmm.
D and L beta hydroxybutyrate with a mixed spread across a mix of electrolytes. Audacious Nutrition Keto Start is one, and that we're working kind of in the background on alcohol-free triesters of ketones that do not use 1,3-butanediol that are far potent and far less toxic. So the liver toxicity of 1,3-butanediol is something that a lot of people don't know about, but we're about to publish some papers on that.
Interesting.
Uh, and I think, so you elevate ketones, lower blood glucose, get a GKI of 1 to 4. Tom's very adamant about 1 to 2, but I think 1 to 4, pretty consistent across different models. And glutamine is a little bit more tricky. You could use EGCG, combine that with sodium phenylbutyrate, which is butanol, and that'll help inhibit glutaminase a little bit, but also lower circulating glutamine to some extent. And and metformin is something that could also be incorporated. Metformin causes a little bit more superoxide production in the cancer cells. We've published on that, and
Like
DCA
complex one, is
too.
that what it's for? Is
Yeah.
that?
Yep. Yeah. My student, Dr. Nathan Ward, he has a lab at Moffitt Cancer Center now. He studied a DCA and metformin in the VMM3 cells that Tom, that was his PhD project. So those things, relatively non-toxic, readily available. And then some of the bigger guns would be like Dawn, and then hyperbaric oxygen is a bit harder to implement. And you mentioned photodynamic therapy. You're probably thinking in the context of the 5-ALA,
Hmm.
5-aminolevulonic acid. And yeah, I was not even thinking in that direction, but that could be, you could photodynamically activate it too. I'm thinking about the patient when the head is not open,
Right.
to do that. So the idea that with the 5-ALA, the artemisinin and the hyperbaric oxygen, you could do that independent of photodynamic activation. But you bring up a good point with photodynamic activation, you could probably wow, you could get like a huge burst.
Yeah, I'm not exactly sure. I'm sure you probably know, but the 5-ALA I think is like either a UV or blue light chromophore, if I'm not mistaken. Do you know what wavelengths it
Mm,
accepts as a photodynamic?
Yeah,
I want to say it's a shorter wavelength.
it's in my research proposal. I should know that, but I don't have that at my fingertips, and I'm hesitant to quote because I'm sure
No
somebody
problem.
will call
Well,
me
the reason
out.
I was going to say is
You
because
can
I think
Google
I
it. Yeah.
think when I think about, yeah, especially like a brain tumor or really any tumor, what makes the most sense to me, I know I'm kind of going off on a little bit tangent here, but what makes the most sense to me is something that's going to accept probably either red or infrared light because it has the most
Yep.
penetrance. So that's why
Mhm.
I think methylene blue honestly is going to be probably a really good
Mhm.
option because it's non-toxic. You can get it. The problem is that when you look at the studies that are done, the millimolar quantities that are able to replicate that would probably have to be IV and probably in the 2 to 4 milligrams per kilogram dosing. So I think that's going to be very difficult for most people to get. But I think that's another direction that you could go as a way to get oxidative stress to a patient, is through
I
photodynamic therapy.
Yeah, I thought about methylene blue too, because I have some of that in the lab, and I was thinking that it could potentially protect the cancer cells. I had a lot of questions about light, and I've talked with what's his name, the light guy, smart
Oh,
neurosurgeon guy.
Dr. Cruz?
Yeah, he's wicked smart. Yeah,
Oh
he's a
yeah, he's genius.
bit of an ego, but the ego is kind of backed up by intelligence, I think, to some extent. But yeah, I've we've conversed a little bit, and we kind of see eye to eye, I think, on most things. And I was going to pick his brain on the the methylene blue. I was also going to pick his brain on some of the photodynamic light things because his knowledge is pretty high on that, and it's a gap in my knowledge, and I need to I think that could be leveraged in a way that we could use that for brain cancer. So you just reminded me to reach out to him because I had some methodological questions
Another
on.
person that could be helpful here who doesn't have the same knowledge. I don't think anybody has the same knowledge that Dr. Cruz has, but Scott Zimmerman is a light engineer. He's done quite a few podcasts recently. He is working with Glenn Jeffrey at the UK, and they're doing a lot of studies. I think he's also working with, oh God, why can't I think of his name? The guy at University of Texas, San Antonio, who's like the melatonin guy. But anyway, so they did a study. They were doing some studies together, and I would say he's probably pretty high level of like understanding
Mhm.
photobiomodulation at minimum. And then
Yep.
if you get into, I don't know about photodynamic therapy, but I bet you he would know a lot about it as well. And
Yeah.
but his expertise is more about like the penetrance of different light frequencies to the body and how it actually scatters via nonlinear optics. Stuff that
Mhm.
again above my pay grade, but like it's still very fascinating because if you look at the literature, you'll think that okay, well, red light only penetrates 1 centimeter into the body, but that's actually not true. Like that's like the way they're measuring it is wrong or something like that, and it really is probably closer to like 10 to 30 centimeters, but it's not linear. It's a non-linear scattering, and there's a bunch of different chromophores that are accepting that, and then it's bouncing around. I don't think
Yeah.
anybody knows exactly what's happening, but I think that red light and infrared light would be your best bet at most places in the body because it has the best penetrance. It's just you got to
Yeah.
find a photosensitive chemical that's able to accept those photons
Yeah.
from that light there. That's why methylene blue is great because I think it's like 650 nanometers or 670 nanometers, which is like right in the red range. And it's kind
Mhm.
of have the most
Yep.
anyway, but yeah, that's pretty cool.
Yeah, I'm a big believer, food is medicine, exercise is medicine, movement is medicine, and light is medicine. So
100%.
I want to do that more. I mean, I've spent my whole PhD career studying, in the dark, studying different porphyrins, tin protoporphyrin 9, zinc protoporphyrin 9, all these various porphyrin molecules that inhibit heme proteins and kind of leveraging that for therapy. So I'd like to revisit and get back to that. Because it can be done in a way that's largely non-toxic and to healthy cells and potentially very toxic if you're really smart about it.
I actually think it could be
Cancer cells.
protective in normal cells for a lot
Oh
of
yeah,
those because like that
yeah,
the whole field of photobiomodulation is about like enhancing mitochondrial function and retrograde signaling. And
yep.
so I think it could be, I don't know if it's the exact same as like ketones and how they have a protective effect against like HBOT or whatnot, but it could be a
Mhm.
protective too.
Yeah, there's overlapping, distinctly different, but also overlapping mechanisms. Yeah, ketones are very protective to the mitochondria. And I didn't even know that until I started working with them. I was mostly focused on other molecules. And when the other molecules were not working, I was doing work with lactate and then the ketones, and I was like, wow, this stuff. And then I just started going down the rabbit hole of the ketogenic diet, and I had to do it because they were just so effective. And I
Mm.
mean, we're talking over, 20 years ago, I started doing this research. But to pivot back a little bit to like, with the diet and just metabolic health, that's why, we have the Metabolic Health Summit, which is like a big summit. And we bring like Thomas Seyfried, he actually got our most audacious investigator award for the last one that we held. And that's why we hold this and we bring basic science researchers, clinicians, and we also bring patients to tell their stories because we
Mhm.
realize that this is like really important. But in regard to metabolic health and the different types of cancer cells, and I think our different types of cancers, I think people maybe don't talk about that as much. We just talk about cancer, right? But I do think there's a lot of nuance there when we say this is going to work for that, or all cancer cells do this. And I think, we do have therapies that are quite effective for everything from leukemias, lymphomas, multiple myeloma, testicular cancer. I know people personally, and I have quite a few people I'm in contact with that are do the standard of care and kind of are relatively curative. Or there are no evidence of disease. But liver cancer is one that is like skyrocketing. I mean, we're talking 3 times higher since 1980. Liver cancer is linked to high BMI, obesity, hemoglobin A1C, HSCRP, I think, or CRP in general, breast, endometrial, colorectal, pancreatic, kidney, esophageal, gallbladder, ovarian, thyroid, bladder cancer and prostate cancer. Like all these, so there's like meta-analysis and reviews on metabolic health and all those cancers I just mentioned, because I just gave like talks on all that. And the evidence is very strong. Actually, it's a little bit debated with prostate cancer, because oddly, like there's a lower risk but like you're more likely to die if you're obese. So if you have prostate cancer, it's like that one cancer that if you're a little bit overweight and you do get prostate cancer, you're less likely to die from prostate cancer, uh, at least in aggressive form. So, but and that's kind of debated. Does it help to be a little bit, leaner. But some cancers, and prostate is that weird one where it's kind of your outcomes are a little bit better if you trend to be a little bit heavier in general. But all the other ones, I think, there's many cancers that are linked pathophysiologically to metabolic health.
100%. As a matter of fact, I will tell you, Dom, that if I have, well, I'll start with this. When COVID was happening, I would diagnose diabetes by someone having a COVID diagnosis. Because what I found was that almost every single patient that ended up getting COVID, especially if they're a younger patient, had
Yeah.
either pre-diabetes or diabetes. Their A1C was above 5.7%, clearly.
Yeah.
Now, with cancer, especially with young people, when they'll come in, this is kind of what motivated me to start this channel was because I was seeing all these freaking patients that were like 30s and like coming in with like abdominal pain, and we'd get a CT of their belly, thinking we'd find constipation or something like basic, and they have like metastatic cancer, right? And so like what I found was that most of those patients also were obese, most, and then almost all of them had both an A1C that was above 5.7% generally, and they had a single digit, low double digit vitamin D levels too. I think those are huge factors that I see in almost every patient that I admit to the hospital, whether they have cancer established already or we find it, on admission, because that's what I do. I do admissions to the hospital. So um, every patient, not every patient, but almost every patient, that's you'll see. And of course, yeah, CRPs, of course. I mean, CRP's a little bit more difficult because CRP, I can kick the wall, and my CRP may go up from the inflammation from that acute injury. Or if I come in
Mhm.
with flu, my CRP is going to be through the roof, but it's hard to really know where it's coming from. But if there's no other obvious reason for it and their CRP's up, then obviously it's going to be. And my understanding is that's associated with poor prognosis too. Is the higher the CRP, the poorer the prognosis it is.
Yeah, and if your gut permeability is high, CRP again kind of goes crazy. A colleague or friend that has like Epstein-Barr like flare-ups, and the CRP was like 50 something, and I was like, well, since I've been on a keto, I've always measured CRP for quite some time, and it's usually like non-detectable. Like it says like ND, or it's like
Mhm.
zero point 0.2 or zero point 0.4, like in that range, usually zero point 0.1 or zero point 0.2. Ever since I've been in ketosis, the only time it's been elevated is when I lived underwater for 10 days in a hypercapnic environment. So I think the CO2 had chronic hypercapnia had that. And also one time I had a bacterial gastroenteritis, maybe viral. I came back from
Mhm.
Southeast Asia, and I was like, oh, okay, be good to do some blood work to see what's going on. And the one day I saw my HSCRP elevated, and it was a reminder that that barrier function in the gut, and just, if you gut permeability,
Mhm.
if that's compromised, your immune system kind of goes haywire. Yeah, I
Yep,
just remember
100%.
my
That's
joints
that's
were
I mean
aching and everything.
that's how they hypothesize that like autoimmunity happens, or a lot of things happen with like that way. 100%. The problem is there's not a real good way to do it. I mean, there's like the zonulin proteins, you can get it in the blood or in the stool, but it's just my understanding is it's not a very good test. And that kind of like also goes back to, are you familiar with Dr. Doug Wallace at the Children's Hospital of Philadelphia?
Oh yeah, yeah, yeah. He's given an IHMC talk. That's
Yeah.
our institute.
So
Yeah, the
I learned
mitochondrial
about
rate.
him through Dr. Cruz because I followed Dr. Cruz for a long
Yeah.
time.
Yep.
And, if you look at his graphics or graphics that are based off of the mitochondrial heteroplasmy type of like situation, you'll see a bioenergetic decline, right?
Mhm.
And of course, like if you look at Seafree's graphics and his papers that he'll have a bioenergetic decline that goes like this. And I think there's 26 genes on the mitochondrial genome, 13 of which code for OXPHOS proteins. And when,
Yep.
and it was funny because Seafried's like, well, they don't have to be hit. They don't have to be completely knocked out. And I'm like, yeah, because they have to just be leaky enough of ROS that leads to, like damage, kind of a snowball of damage, right? Because the mitochondrial DNA are right there in the matrix, right? So it
Yep.
just makes the Wallace's stuff makes a lot of sense to me, and I think I see it dovetailing completely into what Seafried, publishes with the bioenergetic decline and hitting some kind of a threshold. And that mitochondrial heteroplasmy, my understanding is like it's tissue dependent, right? So you could have a mitochondrial heteroplasmy of 10% in one tissue, and other tissues it'd be 60%. And then depending on the disease process, the different heteroplasmy could be different for that too, right? So like you may be,
Mhm.
I don't know, let's say obesity is like, I'm just going to use arbitrary numbers. Like obesity could be like 30%, end stage diabetes could be like 50 or 60%, and then cancer or neurodegenerative diseases could be at a higher level depending on that tissue type. And I
Yeah.
think it's interesting because
Mhm.
that to me like shows that there's no way that oxphos could be really truly happening to any sufficient degree. And that to me like it shows that also is showing that Seafried is really onto something with the treatment. I understand the an ounce of prevention is worth more than a pound of cure. I totally agree. There's not enough time spent to that, but I think where my audience is at is that they're dealing with it. They have it. They have a diagnosis. They had a PET scan. They had a biopsy. They had whatever.
Yeah.
And I think that there's a lot of, and I think this maybe the next question I want to ask you is there's a lot of papers out there that I've read and people send me because it scares the bejesus out of them is they try to they try to say that like a ketogenic diet either grows cancer, makes metastatic cancer worse, whatever. I dug into the papers and what I found was is they were never they were never blocking glucose or glutamine both at the same time, number one. And number two, they were using like super physiologic amounts of either beta hydroxybutyrate or acetyl acetate to show something, right? And I just wanted to know, like, do you think there's like a concerted effort to debunk keto or like make it look like it's not as helpful as it probably is? Or do you think it's just people curious and tinkering around and giving 100 millimoles of acetyl acetate and then having a scary title and like, what
Mhm.
is that about?
I'm not like, I wouldn't say there's a conspiracy theory, but I think there are some labs that will design ketogenic diet trials and design it to fail. And I think that spans across cells, tissues, animal models, and humans. So I've seen that. So I function as a reviewer, an editor, the study section, I review grants from DoD, NIH, and many other. So I've kind of seen that, and I think I could, broadly speaking, we could say anything that, suppresses insulin signaling, it's going to be hard for cancer to grow faster in the context of insulin suppression. I mean, that's with PI3 kinase, mTOR, activating AMPK. So that is like highly unlikely. And if you limit glucose availability, suppress insulin signaling, and then elevate ketones. So it's a good thing you brought up the exogenous ketones. Yeah. And we see this too. And I would caution people chugging ketone esters. When you chug ketone esters, that's actually going to increase insulin. And then an elevation of ketones by taking a ketone ester into that 2, 3, 4, 5 millimolar range, circulating, that actually represents that energy toxicity because you have a lot of energy floating around in the form of ketones that your body is probably not going to use all that. So there's going to be ketonuria. And also, chugging exogenous ketones will spike insulin, but only ketone esters. This does not happen with MCT oil or exogenous ketone salts. So,
Interesting.
interestingly, you can elevate ketones to a delta of 2 millimolar with a ketone ester, and then do it. You can achieve that. That's about the limit of a ketone salt, but you don't want to go higher than that. When you do that, the insulin goes up with the ketone ester, but it does not go up with the ketone electrolyte salt, and it does not go up with MCT. But if you combine the ketone salt with MCT, that's actually the ideal combination because you're delivering exogenous ketones while you're simultaneously stimulating endogenous ketone production with the MCT. And also, the MCT delays gastric absorption in a way that reduces the entry of ketones into circulation, and that prevents it from stimulating the pancreas to release insulin. When you consume a ketone ester, there's a quick, the esterase enzymes work, you get beta hydroxybutyrate, jumps up in the blood, and that triggers a counter-regulatory response, which is an increase in insulin. So these are things that like I think people should know about. Also, 1,3-butanediol, most ketone esters are either 1,3-butanediol by itself, like, HVMN would be one, or delta G or KE4. These are 1,3-butanediol base ketone esters. When you consume them, the 1,3-butanediol is metabolized just like an alcohol. And I worry about that because the liver is like a is an easy target for cancer growth. It's a very fertile ground. And when you consume those types of exogenous ketones, what's happening is that, over a period of time, 1,3-butanediol gets metabolized like ethanol through alcohol dehydrogenase and
Hmm.
then aldehyde dehydrogenase. So you create a beta hydroxybutyrate aldehyde. And if you saturate aldehyde dehydrogenase, then you get a large spike up. That drug disulfiram or what the drug that
Disulfiram.
alcoholics take. Yeah, yeah. So that's actually you inhibit that enzyme, so you get like a lot of aldehyde. I think people need to be cognizant of that, consuming sort of the ketone esters or 1,3-butanediol because you are so what we see biochemical histological changes and a sharp drop in liver ATP when you consume 1,3-butanediol or 1,3-butanediol associated esters. And that could be problematic, especially for older people. And so I encourage people to use an electrolyte, sodium, potassium, magnesium,
Wow.
salt, and then
That's really good to
MCT
know
is good.
because I didn't I did I mean I was reading these papers where it was showing that the esters were spiking ketones higher than the salts, but you're saying that's not beneficial, number one, and number two, there could be a downside of the increasing insulin, which I think is a big downside, but
Yep.
also morphologic changes in the liver at a histology level too. You're saying it's damaging basically.
Yeah, I mean, obviously, like, it's very curious to me when you go on PubMed and you do look at like chronic human studies with 1,3-butanediol or a ketone monoester. Like, there's hundreds of studies, maybe thousands, but there's only two chronic studies with. I know the reason why. I'm not going to say why, but there's federally funded contracts and there's industry interest too that maybe prevent some things. But we are actually going to prevent. We had to notify our IRB because of some toxicity issues. And I'm someone who actually has intellectual property and trials with 1,3-butanediol and ketone esters. So it's coming from someone who has like skin in the game when it comes to using these compounds. But I could say that, yeah, so there's only like and one's a case report, and that's Dr. Mary Newport, her husband, who had Alzheimer's disease, actually used the ketone monoester, 25 grams 3 times per day for Alzheimer's. But he was on some like support supplements too that I think could help the liver, maybe an N-acetylcysteine
Hmm.
and ALA. So if you're gonna use those things, I would actually recommend something like NAC because what happens is you consume 1,3-butanediol, and then I'll just quickly add there's one other RCT, but that only used 25 grams of the butanediol ester. So and that maybe there was like maybe a click up in transaminases, but what I can tell you that if you take a dose of 1,3-butanediol or a 1,3-butanediol ester. And I'm someone who has consumed more of this than any other person on the planet. I can tell you that because, and I did a lot of blood work. We've been tinkering with these things for like 20 years. And 1,3-butanediol is kind of cool. Like you can take it, you get a nice buzz off of it, but you need to be, and I've talked to people who their whole career is studying alcohol toxicity. It is an alcohol. So if you're taking 25 grams, that's like doing a shot. And you need to take 25 grams 3 times a day to maintain therapeutic ketosis. So that's like taking 3 shots. So you're metabolizing that through alcohol dehydrogenase and aldehyde dehydrogenase, and you're generating an aldehyde. So that aldehyde over a protracted period of time is toxic, but also, as you probably know, aldehyde dehydrogenase and alcohol dehydrogenase robs the liver of NAD, right? They
Hmm.
consume NAD. So what happens is, and you can just replicate this, with liver cells, ATP levels drop really quick. So you're de-energizing the liver's energy. You're causing a redox shift. So this is what happens with ketoacidosis, right? Like an alcoholic will consume alcohol, the ethanol, and then they have runaway ketogenesis and they have alcoholic ketoacidosis because it inhibits gluconeogenesis, which is a very energy dependent process that the liver has to maintain. When you consume the alcohol, you de-energize the liver, so it can't do gluconeogenesis. So you get into a very tricky metabolic
Wow,
situation.
that's interesting.
So,
I guess
and
I guess I'm already sold that I'm not going to say ketone esters at this point. But my devil's advocate is alcoholics are also known to like upregulate their, that's why they can build tolerance, right, to
Mhm.
alcohol. So would you not build tolerance to the ketones too? This is the same
Yeah,
mechanism, or?
but you also have to appreciate and understand that like relatively small amounts of alcohol make you susceptible to getting cancer,
Hmm.
right? So you're generating the aldehyde, which can generate DNA. And we have better options, I think. So there's ketone electrolytes. I know there's triesters that are alcohol free that, some people are working on. There might be some companies already. I don't know. We've got like a toolbox of like 30 or 40 different
Wow.
compounds.
Well, salts
So
it is for now. I had
yeah,
one question though. I had someone come at me or, ask me like, is there any literature to suggest that exogenous ketones, will raise blood glucose in any mechanism? Because he was convinced that if he takes ketone salts, his blood glucose is higher on his CGM, and I just didn't know how to explain that.
no, I mean, I don't know. I like to see that because I mean, I've seen a lot of CGMs where, well, it depends so every ketone salt product out there, I'm not going to name names of companies, but usually throws in a lot of other things too.
Mm.
So some amino acids, some leucine, some,
Mhm.
erythritol. What's it sweetened by?
Yeah.
So Keto Start, like Audacious Nutrition, my wife has a company to
I
kind
took
of.
one this morning, so I'm familiar with it. So I'm
Okay,
good.
okay.
Yeah.
Yeah, so that's just like BHB salts and monk fruit, and I think that's it. And then it's got some kind of like, flavor component to it.
One
I
has
mean,
caffeine, one doesn't, though, right?
yeah, the Keto Start Plus black packet has the caffeine in it, and interesting, so that has the D and the L. So that's what actually we ran in
Hmm.
our cancer studies. There's going to be, I think that's the only product out there that has both enantiomers. So there's now a lot of like, I think there's like 50 patents on L. Because I've been talking about it, so I knew people were going to patent it sometime or another. I've been talking about LBHB because it metabolizes about 4 or 5 times slower, and then it retains. You get higher concentrations in the brain. And also the heart in circulation, L is only like 2 or 3%. But if you take the heart out and look at the tissue, there's like 30% L.
Hmm.
So it actually uses it. And the LBHB actually has a profound effect at influencing the afterload. So there's less resistance. So it decreases the afterload by something like 4 or 5 times more than the D, beta hydroxybutyrate.
We're
So
talking about
there's less.
afterload peripheral vascular resistance. We're talking about blood pressure now.
Yes,
Okay.
exactly. So
Okay.
it's like taking, the kink out of a garden hose.
Okay.
And so we are very interested in that property. It tends to relax the endothelium in a way. It
Mhm.
also has like vasodilation effects and anti-inflammatory effects and epigenetic effects. So the L is kind of like think about the L as like beta hydroxybutyrate time release. And then the D, if you consume L and D together, like with the keto start, what happens, you quickly metabolize the D. So that gets burned up as energy, and then the L circulates around for 4 or 5 times longer, and it retains that signaling function and gets into the brain at higher concentrations. And we think the L also inhibits hexokinase a little bit, it has
Interesting.
this blood sugar reducing effect. And that was mentioned to me by Dr. Henri Brunengraber at Case, who had developed some of these racemic compounds. And he used to be the director of the NIH metabolomics program at Case. So he got me kind of interested in L, and we compared all these different things, and we saw some unique properties with the racemic and the L, but a lot of the patents were on like the D and the D enantiomer from NIH and Oxford. So I think like, and that was mostly for warfighter performance. So D will generate about 10% more ATP, but then the L only because it gets metabolized faster. So the L will generate it, but it just takes longer because
Interesting.
the ketolytic
And you're
enzymes are
saying
not
that
there.
almost or maybe I'm missing it. Every other ketone on the market is just D only. It's not D and L both.
Yeah, I don't know of any. I mean, we're talking like every company I can think of, uh, you know, Delta G, HVMN, Kinetics, and all the ketone salts too, I think have pivoted but now these papers are coming out, like the American Heart Association published that paper on the L, and then it's like this miracle compound for, enhancing cardiac output. So that was sent to me when it got out, and that was replicated in other labs. So people just like didn't think about the L because it didn't. We mostly make D, but we do have
Hmm.
a racemase in the tissue to convert it over.
Right.
So now I think the Buck Institute like filed things, and we published a Nature Medicine paper showing the NLRP3 inflammasome is suppressed by L, and that it would happen much more in a much more dramatic way because D gets burned as energy and L hangs
Ah,
around
makes
longer.
sense.
So
I think I've heard you say this. So basically the L is more the signaling molecule like for epigenetics
yeah.
versus the energy piece of it.
But it generates ATP too, surprisingly. If you throw it like in a dish and measure, it's like, you get maybe about 80% of the ATP like that D makes, but then it sticks around longer. And I think about 20% of it ultimately gets interconverted back to D, like slowly. But the important thing is that it stays in circulation longer. It tends to build up in tissues where it has this like remarkable epigenetic therapeutic vasodilation effect. And and to say that it's not the natural form is not correct because we do have it.
Right.
And I could send you a paper after. Maybe you could put it in the show notes. And there's been a couple, yeah, podcasts discussing it. But I guess maybe I was an early adopter of that because I was very resistant to go away from the racemic compounds just because they work so great with cancer,
Hmm.
and then they had an anti-seizure effect too. And then we would compare L and D and didn't really see like much of a difference or sometimes better with the L. So, but like the bandwagon was to jump on D because that's like there was some like patents on that, but we kind of stuck with our guns and
Sure.
but I'm interested in just using what works, right? And but the D for D, if I was going to do exercise, maybe the D would be better, but I feel like D also spikes up insulin a little bit more just because it gets metabolized quicker and it just gets burned. I can tell you if you chug a D ester, kind of what happens is that your levels shoot up really fast. The monitor only measures D. So it's not measuring L could be 2 or 3 times higher, but you're not picking up on the monitor. So
Got
I
it.
think that's really important. It only measures the D enantiomer. Uh, but what happens when you chug, and I've done this many times, so I could tell you, your ketones shoot up really high, and higher is not better, unless you're doing some of the seizure, things that we're doing. It goes up really high, and then you're hyperketonemia, and then you release some insulin, and then the ketones get burned up as fuel within an hour or two later. And at that 2 hour point, you are hypoglycemic and hypoketotic. So your brain, and that can trigger a seizure. So this is the issue with
Is that
using.
only if you're not in like therapeutic ketosis naturally plus adding it in? Or is that because
Yeah, well, if you are in a kind of ketogenic diet and if you chug a lot of it and you release insulin, that will decrease your own endogenous
that
ketone
makes
production
sense.
too. And you can get in a bad situation. People have been hospitalized, because they chug this. So the idea is probably to take it with some carbohydrates, if you're not on a ketogenic diet. So I think, operationally, I think there are good indications for a D ketone ester, but for therapeutic, for the things that we do, the D and the L combination, it's like the best of both worlds.
Right.
So like, I was resistant to jump to the D, and I'm so glad I did, especially since, all these patents are coming up and kind of validating what I always thought and what I
I
knew.
gotcha.
Uh,
And is,
but yeah.
in your opinion, though, I mean, is the better option for a lot of people though, just doing MCT oil only? Because I mean, it's a heck of a lot cheaper, but like, is there clinical benefit to doing, you know, the ketone ester in addition above and beyond a therapeutic ketogenic diet, maybe adding in some MCT oil? Is there benefit to that?
Well, I would take ketone esters off the plate, like if we're talking about cancer, especially the D enantiomer, and then use just
Oh, I
the
don't mean
ketone.
esters. I mean salts. Sorry. I
Well,
mean
salts,
salts.
yeah.
Salts.
So like I like to approach it from the perspective of what can give me the optimal glucose ketone index. And I think some people can achieve that with a modified MCT supplemented ketogenic diet. You have to be very strict with it. But to make that one step more ketogenic, you could do a beta hydroxybutyrate, electrolyte salt, D and L, and then do that, 10 grams 3 times a day would definitely get you there. You could probably do it, maybe even less. But 10 grams 2 or 3 times a day will basically do two things. It'll help bring the glucose down and also bring the ketone levels up. And you might not be even measuring the elevation of ketones. Say if you're measuring a ketone level of 1.2 and you're taking a DL salt, that's highly likely closer to 2, right? Because you have circulating L that's not being picked up
Not being
by
measured.
the Keto Mojo or the Abbott Precision Extra device. We use mass spec and some other things where we can get like total ketones, but and also the salts to interconvert more to acetoacetate. And acetoacetate has we're studying isolated acetoacetate and comparing it to beta hydroxybutyrate. And we're getting like the same ATP levels across, a lot of this is like research that is not published yet. And I think other people have shown that too. I know the guy from Albert Einstein, Eugene Fine, did some studies with acetoacetate and showed it had remarkable anti-cancer effect across all different cell lines. And I think acetoacetate is, equally important. And the salts, the beta hydroxybutyrate salt, interestingly interconvert to circulating acetoacetate to quite a bit. Like it seems like a third of it. And you can actually pick that up as acetoacetate with a urine, or you can also pick it up as breath acetone. So acetoacetate will spontaneously decarboxylate to acetone, and then you can pick that up on breath acetone meters. If you chug a lot of 1,3-butane-diol and blow into a breath acetone meter, you don't get anything. But if you take the salts and get the same level of BHB, then the acetone measurements. We've done that, and I have a number of people kind of sent me their data on that.
That's
So
interesting.
there's a lot of interesting dynamics we still have to understand about how ketones interconvert from BHB, but BHB needs to be converted to acetoacetate to be used as energy. There's
That's
a
a great
problem.
segue, Doc.
Yeah.
So I think there is a lot of fear within this kind of community. And, Dr. Seyfried's done a lot of work on this, and I'm not sure if there were papers that you were associated with. I mean, they're on ketones, so they may have been, but can can ketones be burned by non-mitochondrial containing cells or cells that are don't have functional oxphos? Is there a way that they can be fermented through mitochondrial substrate phosphorylation in any way that you're aware of? I hope you enjoyed this first half of the interview with Professor Dom D'Agostino. I know that I did. I learned a ton. I'm gonna have to listen to this a couple of times over again to see if I can pick up some other details that I missed. If you like videos like this, if you like the fact that I'm having these expert guests on to help us move our understanding forward, please like, share, and subscribe. And until next time.